So I hope you guys are feeling awake because the first half of our day is going to be science-based. Give it up for the scientists. So we're going to hear research updates from several of the labs that are studying KDM5C. However, since some of us in the audience are not geneticists or neuroscientists, we are going to start with a session that we like to call Genetics 101 to ground us in some of the scientific language and the basic genetics of KDM5C variants. So I'd like to introduce Dr. Catherine or Katie Keegan, a professor of pediatrics and human genetics at the University of Michigan. Please welcome Dr. Keegan.
Thank you, everyone. That first talk was really hard to follow. I really appreciate Adrienne coming and telling her story. And that's, really for me as a physician and researcher, that's the reason why we come, is to hear all of your stories. So we really appreciate that. And I'd also like to thank the KARES board for the invitation to come and speak. This is really great. We're going to switch gears and talk about genetics. So hopefully everybody is ready for this. Okay. So the outline for my talk is first to review DNA, chromosomes, and genes. And then we'll talk about different types of genetic tests. We'll talk about what are specific or different types of genetic changes, and then how to read a genetic test result. So we'll first start out with DNA. Let's see if I can, here we go. Okay, so this is a DNA molecule. DNA is a double helix. Everybody's heard about the double helix, formed by base pairs attached to a sugar phosphate backbone. So we've all heard about the different base pairs. There's adenine, thymine, guanine, and cytosine, or ATGC, and that makes up the DNA. So next are chromosomes. So chromosomes are structures made of DNA. This is just a little cartoon of a chromosome. Chromosomes have what we call a P arm, or a short arm, and a Q arm, which is the long arm. And chromosomes are composed of DNA. And then there are histone proteins that sort of hold all the DNA together and help compact it into the structure that's the chromosome. So each cell in our body has 46 chromosomes. This is something we call a karyotype in genetics. And so all the chromosomes are laid out in a particular order. So they come in pairs, and they're numbered from chromosome number 1 to chromosome number 22. Those are called the autosomes. Those are the same in men and women. And then there are the sex chromosomes. So the X and the Y chromosome. Boys typically have an X and a Y. Girls typically have 2 X chromosomes. And as all of you know, the KDM5C gene is on the X chromosome. So next, chromosomes contain our genes. So again, this is the structure that is a chromosome. It's made up of DNA. And then we can identify specific regions of the chromosome that contain our genes. So this is just a cartoon of what a gene might look like. So there are lots of different analogies that people use to talk about genes and chromosomes. One of the ones that I kind of like a lot when I'm explaining these concepts to patients is that they're the cookbooks, the recipes, and the food. And so the cookbooks kind of represent the chromosomes, the recipes represent the genes, and then the food represents the proteins. Other people use things like a set of encyclopedias with different chapters representing the genes and the words in the chapters representing the function of that gene. So again, the food is what we call, the proteins, and that's made up of, that's basically proteins perform different functions in the body to specify how we grow and develop. So proteins are contained of amino acids, and then they're sort of linked together in a chain, and they have, different amino acids. So for example, in genetics, we know that Phe means phenylalanine, Lu means leucine, serine, cysteine, and that sequence sort of makes up the protein and specifies how it does its work. So talking about different types of genetic tests, some of you may have heard of some of these, but not all of them, but I'll just sort of run through them real quickly. So first is the chromosomal microarray, then we have gene panels, then we have whole exome sequencing and whole genome sequencing. So we'll talk a little bit about these different types. So this is just a chromosomal microarray. So I like to describe this as sort of a screening of the chromosomes for missing or extra pieces of genetic material that might contain a number of genes. So they're larger pieces and just surveying all the chromosomes. And so this is just a picture of what that data looks like, where this shows a normal chromosomal microarray, and this blue line just sort of represents to us when we're looking at it that there's the normal amount of genetic material. This is just an example of a deletion of part of chromosome 4. And so you can see this little blue line here is a little bit lower. And so when we're looking at that in the laboratory, we know, okay, that's a missing part of chromosome 4. And then on the other hand, this one shows that there's an extra, so this line is sort of thicker, and then when we're looking at this in the lab, we can tell it's a little bit higher, and down here we can see there's an extra line, and so that tells us that there's a whole extra chromosome, so that would be trisomy 13. So again, this allows us to say whether there's missing or extra genetic material. But what it doesn't do is actually look at the sequence of the genes. So for that, we use other tests called, there's a gene panel, which basically does a spell check or looks at the sequence of DNA. But for a gene panel, we're only looking at a focused number of genes. So there's what we call a neurodevelopmental or an epilepsy gene panel that has a collection of genes that are all known to be related to specific features that a child has. And so again, that performs a spell check and we can see differences in the DNA. But because it's a focused collection, it doesn't look at all the genetic material. So we have other tests that will do that now. And so the first one is called whole exome sequencing. So that's a spell check of all 20,000 genes. So we know that we have about 20,000 genes. And rather than focusing on a small number of genes, this one actually looks at all the genetic material. But the way that this is a little bit different from the next test that I'm going to talk about is so the exomes is the protein coding part of the genome, or it's the part that performs it's the functional part of the genome. And so that only represents about 1 to 2% of the DNA. And so another test that we have now is called whole genome sequencing. And so that also does a spell check of all 20,000 genes, but it also looks at both the coding and the non-coding parts of the genome. And that has some benefits over the exome sequencing. The problem is we don't know or understand what a large part of the non-coding part of the genome does. The other issue with the whole genome sequencing is that a lot of insurance companies are not covering it yet, whereas they'll cover the exome sequencing, but some people may have had genome sequencing. Okay, now we're going to talk about different types of genetic variants. So, and maybe some of you have heard this when you got your genetic test reports, but the changes that I'm going to discuss are missense, nonsense, there could be insertions, deletions, and then there's something called a frame shift. So this slide talks about the different types of genetic changes. And so this uses an analogy of the chromosomes are like the chapters in a book and the genes are like the sentences. And this is kind of an old slide, so it uses mutations, but now we typically use the word variants instead of mutations. But mutations are like misspelled words or variants, and so they disrupt the meaning of the sentence. So if you have the word red, you can say the car was red. That red can change to RDB, which doesn't really make sense. Or you can, if you miss the word car, it just says the was red, and that doesn't make sense either. So talking about the different types of changes, there can be what we call missense changes, and that changes one word or one letter. So as an example, if you had the car was red, if you changed a word and said the car was hat, that doesn't make sense. If you changed a letter and said the car was RDD instead of red, again, that changes the meaning of that sentence. Then the nonsense mutations, that's where the sentence ends early. So instead of the car was red, you just have the car. And so there are missing instructions, and that also changes the meaning of that sentence because it doesn't make sense anymore. Then if you have an insertion, if you add a word or a letter, again, you add the car was red, red, or the car was red. Again, that changes the meaning of the sentence and it doesn't convey the correct meaning anymore. And then similarly with a deletion, the car was red. Then if you delete car, you just have the was red, or the AR was read. And again, that changes the meaning. So these are the types of changes that we can see in the genetic material. And then talking about a frame shift, so that's a little more complicated. So when we have the DNA, it comes in like 3 DNA base pairs will specify 1 amino acid in the protein. So here we can see up here are the DNA base pairs, here's the amino acids, and this sequence of DNA here specifies these particular amino acids. But what happens is with a frame shift and that's caused by either a deletion or missing 1 base pair or adding 1 base pair. And if you do that, it throws off that reading frame or the 3 base pairs, and it results in the production of a different amino acid. So again, it sort of shifts the way that the DNA is read into the amino acids. And so that's called a frame shift. Okay, so now I'm going to talk about how you would read your genetic test report. And so I have an example of a genetic test report here. This is from GeneDx, which is a laboratory that a lot of doctors use to do exome sequencing. Talks about the test requested was exome DX, which is basically whole exome sequencing. The doctor provides the clinical information about the patient, and then the results, they talk about causative gene variants, variants in genes that are possibly associated. And then a lot of you have heard of ACMG secondary findings, maybe if you had exome sequencing. So that's a list of roughly 70 to 80 genes, depending on when you had the testing done, that the American College of Medical Genetics recommends just testing because there are things like risk for early cancer or early onset heart disease and things which are actionable. So some people can opt into that, some people opt out. But it will tell you the secondary finding results on the test report. This actually happens to be what we call a reanalysis, where the labs will typically actually review the test results again after a couple of years. And so in this particular case, it was saying that the reported variants had no change in their classification. So I'm going to draw your attention here, which I'm going to show you on the next slide, but here's where the KDM5C variant was reported. Okay, so this is just a blow up of that. So we have the gene KDM5C, the disease KDM5C related disorder. We have the mode of inheritance. So again, it's on the X chromosome. This is the specific change. So this is C means the cDNA, or that's the DNA sequence, and it's telling us the specific position of the DNA. So 3344, it's telling us that a G base pair was changed to an A. And then it tells us the protein sequence. So the P means the protein. R is the specific amino acid. So it happens to be arginine here at the 1115 position, and that was changed to an H or a histidine. Hemizygous just means that the change was only found in 1 copy. So for X chromosomes, males are typically called hemizygous because they don't have a second X chromosome. And it tells us that that change was inherited from the mother. And then the laboratory is classifying this as a variant of uncertain significance. And we're going to talk about that more in the next slide. So these are all changes that I just pulled from our registry. These are all changes in KDM5C. And so just thought we would sort of run through them again. We start out with a C at the position of the DNA sequence, where that is specifically, what the change is, what the change in the protein is. And because we're just changing 1 amino acid here, so we're substituting this L or leucine for a proline, that's called a missense mutation. This change, again, the cDNA, it's telling us what the change is. And then in the protein, it's saying P, it's changing this G, or it's a glycine, to an X. And that X means that it was what we call a stop codon. So that stops the protein. So that's a nonsense change where the protein just ends. Then this one, the cDNA, it says del T, so that's missing the T sequence from that one spot at 1501. Tells us what the effect is on the protein. So the protein change is from something called tryptophan to a glycine, and then it has this FSX48. So what does that mean, right? So what that means is the FS means frame shift. Then what happens is, because that protein sequence is thrown off, like I showed you in the one slide, the X means that it'll stop after 48 additional amino acids. So that's a deletion variant, and it causes a frame shift. So here is sort of is the opposite. So you have a duplication or an extra G at this position, tells you the protein change. This is E, or a glutamic acid, changing to a glycine at the 1405 position. Again, the FS means frame shift. And then X, after 11 additional amino acids, it stops. So this is an insertion mutation, which causes a frame shift. And then the last one is just another nonsense mutation where at this position, there's a cysteine that gets changed to a stop. So that protein stops. So talking about different types of genetic variants. So all those changes that I just showed you, they're classified as a specific type of variant. So this is sort of a spectrum here, ranging from benign or likely benign, and those would be typically called negative test results, all the way to a positive, which would be a likely pathogenic or a pathogenic variant. And then there's this whole thing in the middle called the uncertain test result, or the variant of uncertain significance. And I know this really drives families crazy sometimes, but it's just the way that the lab classifies those changes. So the benign or likely benign are changes that are not thought to cause a particular disease or condition. The pathogenic, on the other hand, are changes that are thought to change the function of the protein, and so they're classified as disease-causing. And then the variant of uncertain significance is in the middle, where we don't have enough information to say either it's pathogenic or it's benign. So it's just the way that the laboratory classifies it, but that's what that means. All right, so going back to analogies, we think about recipes. So this is a recipe for vanilla cupcakes, and you can see that it has 2 eggs. But if you add an extra G in there, you still kind of know what that means, and so you're able to make the cupcakes, okay? So that would be classified as more of a benign change. On the other hand, if it said 22 eggs, that would cause more problems, right? And so you get something that looked really, not edible. And so that would be more like a pathogenic change, okay? Another way to think about this is another way of explaining changes. So look at the word theater up here. Okay, so we all recognize that as saying theater, and we know what that means. But in Great Britain, they'd say theater with an RE, right? So that's changed the wording, but we all know what that means. So that would be more of a benign change. Whereas if we replace the T with a C, that's cheater, and that's a totally different word. So that would be more like a pathogenic change, okay? All right, so in terms of interpreting the genetic test results, so your doctor will receive the genetic test results, and they'll try to interpret them in context of the child's features. And so if the results show a pathogenic or a likely pathogenic change in KDM5C, that would result in that diagnosis. So your child would have a KDM5C related disorder based on that change. So the VUS means that change is uncertain, but your doctor can say, well, gosh, yeah, the lab said that this is a VUS, but I think that this child fits KDM5C perfectly. And so I'm going to say that that's more of a pathogenic change, okay? And so the doctor can really sort of specify whether or not they think that diagnosis fits for that child, even with a VUS. But in terms of VUSs, so if parents participate in research studies for their child in a laboratory, that can actually help clarify whether that change is more likely to be a pathogenic change versus a benign change. And so some parents will choose to do things like that as well. But that's really the best way to classify or clarify whether a change of the VUS is benign or it's pathogenic, is to have research studies performed. And so how does genetic testing help? Well, it really, all of you have had genetic testing for your children, so I don't have to really explain the benefits, but basically it provides an explanation for all of the concerns that your child has. So, and what we like to say in genetics is it really ends that diagnostic odyssey. A lot of people, they know there's something wrong with their child. They spend a long time trying to get a diagnosis, and being able to find a genetic diagnosis really ends that diagnostic odyssey. It also may help identify future medical problems that need to be screened for. So if we knew that, epilepsy was a concern for KDM5C, it's not in a lot of patients, but if parents see symptoms that they're concerned for regarding seizures, they want to get in touch with a neurologist. It can also give families information about other children with the same diagnosis from either the medical literature or from Facebook groups, and can help families in that way. And I think that this entire conference, the next point is that it really facilitates patient and family support. So, creating Facebook groups and support organizations like KARES really provides that education and support and community for families. And then finally, it helps understand the potential impact on other family members. And so let's talk a little bit about inheritance. And this has probably been explained, but we all know that KDM5C is on the X chromosome. So we're talking about X-linked inheritance. I'm really just going to focus on this side. So typically what we see with KDM5C is that the father is unaffected. So he has his X chromosome and a typical sequence for KDM5C that is functioning typically. Mother can be a carrier, so she has a variant change in KDM5C, but the other copy is typical. So when this couple has children, they can have a daughter who's completely unaffected, so got that copy from dad, got the typical copy from mom. They can also have an unaffected son, so they get the X chromosome from dad, or I'm sorry, the Y chromosome always comes from dad. Sorry about that. So the Y chromosome coming from dad, the X chromosome with the typical copy of KDM5C coming from mom. And then the other possibilities are that there's a daughter who's heterozygous. So she gets the X chromosome from her dad with a typical copy, and then she gets the variant copy from her mom. We all know that there's a range of features in girls. So some girls don't have any symptoms at all. Some girls have some mild symptoms, and some girls have more severe symptoms. And I would say that we really don't understand the full effect of how that works in girls at this point. And then for the boys, so the boys, again, inherit their Y chromosome from their dad. And then this boy inherited the variant copy of KDM5C from his mom, and so he would be affected. Now, there are occasions where parents are unaffected, and that change occurs either in the egg or the sperm cell. And so that's what we call de novo, or a new change that occurs that may result in either an affected son or an affected daughter, and the parents don't have those changes. And so for that case, the parents would have a low risk of passing that on or having another child with the same change, but that affected child, again, would follow this inheritance pattern. Okay. So just acknowledging some collaborators on the KDM5C work that we've been doing. You're going to hear from some of them today. Some people that I work with, I stole slides from them, so I wanted to acknowledge them for helping out with the slides. And then financial support, the Farahi Research Fund, and then KARES for subsidize or paying for us to come to this meeting, which is great. All right, any questions? I don't know how much time I took. Yeah.
Wait, I'll come to you with the mic so that we pick up audio for our people on Zoom as well.
So on our daughter's genetic report, it shows a letter, and then it has a greater than sign, and then another letter.
Yeah.
So what does that one?
Yeah, that one was, let me just show you, because I talked about that. Let's see. Let's pull that one up.
We're all going to get our individual genetic
That's
consults
okay.
later.
Hey, that's what I'm here for. All right, so that's like this. So it's probably talking about the DNA sequence, and then it'll give you the position, and that's the base pair. So this changes a cytosine to a thymine. Okay, so it's talking about the DNA change. There's someone in the back.
Wait, everybody has mics.
Okay, so we come from Canada, and in Quebec, when my grandson was diagnosed, the doctor said that. So they told us that the parents doesn't have to be affected to get for my son to have it. So with your last slide,
Yeah.
so it means that one of the parents has to have. No. It can happen randomly.
It could be a new change that occurred either in the egg or the sperm cell.
Okay.
So that, yes, is that what you mean?
Yeah, because
uh-huh.
on your last slide, I was
Yeah, yeah, yeah.
understanding that
Yeah.
one of the parents has to have something mixed for the kids to have it, but it can arrive randomly. Okay, so you're good. Yes, it's Okay, thank you.
I should have shown a slide with de novo. I didn't have one of those. Sorry about that.
If you can go back to the,
Yeah. Back to
one?
the other one. Yep.
yeah, I think someone already said this is like our genetic consult here. Um,
Hey,
For the,
no problem.
so the numbers, letters, and then the P, after the P, we've seen some tests that have a question mark, and that's it.
That means it's unclear how that affects the protein. Yeah,
Okay.
so, for example, if it's like an intron variant or a splicing, if it's a splicing change, I didn't really go over that, but it might say question mark because they can't tell how it affects the protein.
Okay, thank you.
Mhm.
So one up here.
So on the first one, the makes
Mhm.
sense one,
Yes,
but it says C greater than T,
it just says it's C changing to a T.
that's the amino acid,
No, that's the DNA,
the DNA.
the DNA.
And then the
Mhm.
P and the L, that's the protein.
The P and the L, so that is the amino acid change. So, that's from, and I didn't really put up the code for the amino acids, but P is proline and then L is leucine. So that just means that instead of a proline at that site, there's a leucine. Those are just two different, so it's swapping out one amino acid for the other. Sometimes those changes don't affect the function of the protein, but sometimes they do.
The C and the T, it's the DNA.
That's the DNA change.
And what is C stands for?
I'm sorry.
What is C stands for?
Oh, C is the cytosine?
Uh-uh.
Yeah. There's some back there. Oh,
Okay.
yeah.
I'll go here, here, and there.
Hi, so I have a mutation as well. It's not, obviously, it's not between KDM5C.
Yeah, uh-huh.
But could that possibly affect a de novo?
Probably not. I don't think
Okay,
so.
like mine
Yeah.
affects the cellular lifespan of,
It's new.
right, so,
Uh-huh.
so I was wondering if it.
I don't
if it
think
might
so. Are you
actually-
saying like, could that have caused the genetic change?
Right, in the sperm?
Yeah, probably not.
Okay, I was just
Yeah,
curious. about
yeah,
that.
yeah.
Is
Mhm.
there someone else in Canada?
I think there were some other questions too. Yeah, right
Right,
around the.
Making the
other
rounds,
side of
sorry.
Thanks, Katie. I have two questions. One is a simple one, and that is, should our family members that have this genetic condition, should we refer to them as having a genetic mutation or a genetic variant?
Right, so.
And
yeah.
then the second question
Yeah.
is more specific. Is there a certain part of the gene that is more common to have severe phenotypes or less severe phenotypes?
Yeah, those are great questions. So in answer to the first question, mutation is kind of the outdated genetic term. So now we would all typically use variant.
Okay.
Because a variant, there can be a variant that's not, it doesn't change anything like one of those benign variants, and that wouldn't be called a mutation. So, now in the field of genetics, we use genetic variant.
Okay, thanks.
And then the second question was about, that's what we call genotype phenotype correlations. I don't know, Shigeki, are you talking about that in your talk at all? Okay. Oh, Julie will. Okay. So
There's
I
a
think
lot
you'll
more
hear more about
science.
that later. Yeah. Okay. Yeah. And I don't know if Kristen put that in our registry talk, but anyway, we'll talk about that more.
So I was looking at my mutation and it's in the exon. So what is the difference between having a mutation in the exon versus the intron? And how does that impact phenotype of the condition?
Yeah. So changes in the exon, that's the protein coding part. So that's the functional part. So the changes in exons are all of these that are shown up here. But those changes, like I showed you on the other slide, I'll just show this. even changes in the exon can either be benign or pathogenic. Now, changes in the introns that's not the functional part, but some of those can affect the splicing, which means the way the gene is put together. It's the way it's strung together, or the protein is strung together. And so some of those changes can cause a pathogenic effect, but most of the changes in the introns would typically be thought of as benign.
Oh, wait for the mic, sorry. It's for Zoom.
Sorry, a lot of these variants, like you said, some of them could be like missense variants, right? So that's like a single base pair.
Yes.
So would CRISPR be a good way of treating that?
So with CRISPR, you can make the change back to the normal sequence. You'd have to think about what cells you would do that in, how you would get those cells into the patient. So for example, for things like sickle cell disease that affect the blood, they're now taking out those cells from patients and making that change and putting them back in, and it's easier because it's blood. And that is actually a new therapy for sickle cell disease. So for a condition that affects a lot of different cells in the body, particularly in the nervous system, it's a little more complicated. And plus, like at the time period of development when the change in the gene causes the effects that you see later. So, but theoretically, you can make a missense change back to the normal sequence using CRISPR.
So would you say that the big harder right now is delivery?
Yes.
Okay, and then sorry, one more question. Would you recommend like families to have a gene expression, so like mRNA sequencing gene expression, because KDM5C, of course, is a transcription regulator?
Yeah, that's a great question. So Dr. Awasi may talk about that a little bit, but yeah, and the other thing to think about when you're looking at gene expression changes is what tissue you look at, right? Because, and I think KDM5C is probably expressed in all cells, and so you might see, if you're taking a blood sample to look at gene expression, that might be different from if you were, obviously nobody's going to do a brain biopsy, but it's going to be different from what you might see in nervous system tissue. I think that doing RNA-seq or, looking at expression changes on a research basis is a really interesting potential opportunity to learn more about KDM5C.
All right, we got time for one or two more.
Okay, quick question. I was wondering if my questions are very specific to my son and his report and helping me understand what I'm reading a little bit better. Is there a way that I can follow up with you after this? Sure. or contact information or?
Yeah. I actually didn't put my email on, but I can have you send out my email address. Yeah.
And anything that you guys need post-conference, you can always email info@kares.foundation and we'll get you connected.
Would it be possible for us all to have a handout of all your slides, but also could you include the one with the de novo? Because that's quite important, because that was totally missed out.
I'll try to fix that, and I'll send a copy to.
Within the world we live in, everybody thinks it's inherited.
Yes,
sometimes it is, and sometimes it isn't.
Yes,
and I think it'd be helpful to be able to show.
Yeah,
people.
for sure. Yes,
we feel a bit.
No, I appreciate that. I should have included that.
You did say it, but we all tend to just look at slides. I think, when there's things on the screen.
Sure. Yeah.
Okay, last one.
I just wondered if there's a difference in the way a carrier's gene looks versus a variant.
So a carrier, that term in genetics is typically used for someone who carries a genetic change, but they don't have any effects or features of that condition. So in particular for things that are on the X chromosome, in the past, we would have said that females are carriers, but we know with KDM5C, some women and females are carriers because they have the change and they don't have any symptoms. But then we know for KDM5C that other girls have symptoms. And so they would still be carriers of that genetic change, but they would also be affected. Does that make sense? Yeah. Okay, thanks. Okay.